Yes, in this lecture we are going to cover or study something about enzyme kinetics. I think in the previous lecture I completed microbial kinetics. I showed some empirical relationship like that might look like first order expression. I covered monod kinetics. I talked about the limiting conditions of monod kinetics. Let me cancel this one. So I derived the expressions for monod kinetics, and I also showed applied few conditions like when your substrate concentration is much higher than your one of your monod constant. I also stressed about the importance of mu max and also the units for Km and also the other kinetic constant involved in the monod expression. So in this lecture we are going to cover enzyme kinetics. Yeah. So enzyme kinetics is the some of the equations that we are going to derive now. It might resemble to resemble monod expression or monod kinetics, but it will have a completely different meaning. Yeah. So I'm going to start again with some basics. So what happens in a biological reaction? So I already showed some different easiest ways to write a biological reaction. So one is your microorganism, and then your substrate. So they consume your substrate and they produce new products. Yeah? Products plus some energy because you're breaking some molecules. So I hope you all remember these basics. And exactly, if you want to say what happens in a biological reaction, say your living cells like bacteria, what they do is like they consume your substrate, or if you in some and then they grow or they replicate, or if you want you can call they reproduce, and then they transform your substrate into new products. Yeah, so this is the basic concept you should remember whenever you think about your microbial reaction. Yeah, so something is getting consumed and something is getting converted into new products and something is replicating inside the reactor. But this is like a general observation. You can come to this conclusion just by looking at the whole process and by monitoring your reactor. Like you can monitor your substrate concentration, you can monitor your microbial density, and from that you can write some simple expression like this, or you can have a general understanding that like living cells can convert your substrate into product. But if we go a bit deep inside, or if you have the ability to, or if you have some understanding from the biology, what really happens is these living cells, these microorganisms, so they produce something we call them as enzymes. So this enzyme, so enzyme is the guy who do the job. So it converts your substrate, it act on your substrate and then break your substrate into products, yeah, which has some commercial value. So enzyme is like the key ingredient. So if your live cell, if there is no active enzyme, so if you give a substrate, it will never be, you can never convert your substrate into products. So the enzyme which is in your live cell, he's the guy who is doing this job. So the enzyme acts on your substrate and break your substrate and then convert them into new products. Yeah? So this is some understanding you should have in your mind before we are going to study this topic. Yeah? Enzyme kinetics is a very tough topic. In fact, yeah? You should really understand the whole concept before we are going to do some mathematical derivations relevant to this topic. Yeah. I'm just checking my notes because I'm going to follow the notes. So I will upload the PowerPoint and I will try to keep the same order which is in the PowerPoint. Few things you should remember. I think I already covered in lecture 2 or 3. I showed like you start from molecular precursors like carbon dioxide, like water and nitrogen, and from there you make your amino acids. From amino acids you get your amino acids. From they combine to form proteins, and proteins, sorry, they combine to form peptides, and they combine to form proteins. Yeah? So these are your enzymes. So proteins, so these are your enzymes, and from proteins you go on to other higher level molecular assemblies. We call them as supramolecular assemblies. You should go to lecture 2 or 3, I'm not sure which one, and then we go to organelles or parts of the body. If you want, you can call them, and from there you go to cells. So cells are the like particles, and then enzymes are more like molecules. So for cells we can measure their weight. So how much like mass of cells in unit volume of your reactor. For enzymes we can define mass of enzymes, but and we can also get some information about their molecular weight. So we have a definite structure for enzyme. Yeah? Live cells, we don't know what's the like we cannot define a molecular weight. So whenever we say like we added 1 gram of your live cells into your reactor, you always have to stick to these units, like in terms of weight. Yeah? But whereas if you have an enzyme, you say 1 gram of enzyme, you can convert them into moles, because we know the molecular weight of enzyme. So gram of enzyme divided by molecular weight of enzyme, so that will give you moles of enzyme. So try to remember, enzymes are molecules. Enzymes are molecules, and molecules have definite structure and shape, composed by many number of atoms. And enzymes are proteins, and enzymes do the job of converting your substrate to products. So this at the moment we are just covering the basics of enzymes. Yeah? And remember, this is bioprocess engineering, so we don't have to go to the too much into to the biology part, but we should have some basic understanding about what are enzymes. Yeah, like at least you should know enzymes are molecules. Yeah, because it might look basic, because but once we are going to cover more and more on this particular topic, you will know why you have to understand enzymes are molecules, because and it has molecules mean it have a definite structure, and if you have a definite structure, which mean there are some functional groups and there may be some active sites, and so that's why I'm talking, just stressing about this concept. Enzymes are molecules, which mean they have a definite structure, or if you want you can call molecular structure. They have many number of atoms. All these atoms combine to form a big molecules, and I think and these molecules are not a small molecules. No, they're really large molecules, like ranging from 10 to the power of 4 to 10 to the power of 9 gram moles. Yeah? They are, if you want, like by this enzymologies, they always express in terms of Daltons, this biological thing. Yeah? But anyway, both are same, Daltons and gram mole. Both are same. So always remember, so the units of enzyme is gram per moles. Sorry, not unit of enzyme, sorry, their molecular weight typically range from 10 to the power of 4 and 10 to the power of 9. So this is a really big number, yeah. So from here to such a big number. So they are not just molecules, they are also larger molecules, enzymes. So these are the things you have to remember. And then, and what are enzymes? Enzymes, so in a biological reaction, you can call enzyme as catalysts. And why we call them as catalysts? Because it possesses some catalytic property. Yeah. So enzymes are proteins. They are larger molecules. They have a definite molecular structure, and they are catalysts because they have some catalytic properties. So they can convert your substrate into product. And why? So substrate and enzyme, and you convert them into products. So we call them as catalysts because they just participate in the reaction and they speed up your reaction and convert your substrate into products, but will not involve in the reaction. So you will get product plus enzyme again, yeah. So they will just accelerate your process. So they will help you to convert your substrate into products very faster, but it will not involve in the any chemical reaction, yeah? So these are some basic things you should know about enzymes, and few other things like let me go back to my notes. I don't know which page I am. Yeah, and then you can go to, as I said, you have some cells and you have some active units. You can call them as enzymes. If you want, you can cut the cells and then take the enzymes out. So you can use them as a you can call them as enzymes, and then you can take a substrate and then you can add your enzymes and then convert them into products, yeah. So if you want, you can call cells, this one, the whole unit, as factories to produce enzymes. I'm just covering randomly the basics of enzymes before going into enzyme kinetics. So what does that mean? Like we can disintegrate a cell and then we can extract a cell, sorry, and extract the active unit, which is called enzyme, and which have some catalytic property. So that's why we are calling cells as like a factory or a unit where enzymes can be produced. And then catalyst I already mentioned. So what they can do is like they can take your reactor and convert it into products, and it won't consume your products either once you form some product into the in the reactor, or they and neither they participate in the biological reactions involved, yeah. So, and one more thing, it's very important, yeah. So your cells, the whole unit, see, the whole unit, your cell, this is a live material, yeah. So they can breathe and then they can replicate, but the active ingredient which is inside, they're not a live material or a live product. So which means your enzymes, once you cut the cell and you take the enzymes out, this is not a live material. So you have to remember this. So it's not a live material. And then I think my handwriting is really bad. I will try to write it very clean from now. So enzymes are molecules with molecular weight 10 to the power of 4 to 10 to the power of 9 grams per mole, they are catalysts, they are proteins, they are not live materials. So these are the things we covered so far, and they have a, since they are molecules, they have a definite molecular structure. This is stating the obvious, yeah. They have a molecular structure, and since this is a live material, they cannot digest the product. They cannot digest your reactants. They cannot digest, or they cannot eat, yeah, if you want to call in easy. They cannot eat your substrate, which means they cannot digest, but what they can do, they can break some complex molecules into simpler compounds. So they can break your complex molecules into simpler compounds, yeah. So I think I'm going to, the PowerPoint slide which I'm going to upload in the Sulis, I gave some example. It's a work from Japanese scientists. They published their work in 2016. So what they did is like they tried a different bacteria from a bottle recycling plant. So they wanted to degrade the plastic, yeah. So and they found one cell. It's called I don't know how to pronounce, Idonella sacchaensis 201F6, yeah. They found that this particular microorganism, they can or bacteria, they can eat the plastic, they can digest the plastic. So they found that this particular organism, they can digest the single use drink bottles. So what does that mean? And they wanted to know how this is happening, and they figured it out. So what this bacteria do, it generates or it secretes an enzyme, yeah, or if you want, it's a type of protein, as I said. It secretes an enzyme called protease. So this enzyme, it can split the chemical bonds esters in PET. So this is a polyethylene terephthalate. So this is how the one they use to make the plastic bottles. So this protease, this particular enzyme, they can break the chemical bonds of in the polyethylene terephthalate, and then they break them into smaller molecules. And these smaller molecules, this will act like a food for the microorganism. They can eat these smaller molecules and they can digest. So this is a very good example to differentiate between the job of a live bacteria and the job of an enzyme. So what we do is like we take or how to put it in easy way, like you take a substrate, you add some live material, and this live material secretes some enzyme, and this enzyme can break those heavy complex molecules into smaller compounds, and that can be easily digested by the live cells. So that's how it works. So if you want to do the same job, you can break the cells, and then you can take this particular enzyme out of the live cell, and then you can use this particular enzyme to break PET, polyethylene terephthalate, into smaller compounds. So hope you are all following this one. So So this is the difference between a live cell and an enzyme. Yeah, so one thing you should remember always is like they cannot eat, I mean enzymes, they cannot eat, they cannot digest, but they know to break complex molecules into simpler compounds, or these simpler compounds act like a food for bacteria, and bacteria can consume those simpler compounds and then decompose them. Yeah, so this is a cool way to understand this topic. And then and few things that you should know about biological reactions. So if you want points to remember, or you should have some basic understanding, like any chemical reaction, biological reaction, or like how do you call, like the outcome or the kinetics of the biological reaction. Say imagine you have time, and you say you take your substrate in your reactor, and then you add some enzyme, and then you convert your substrate into products. Yeah? So and just imagine you have some probe to monitor your product concentration, and if you plot, you will get something like that. So what it says is like your product concentration is increasing with respect to time. Yeah, so and if you start from say 100 grams per liter of your substrate, or 200 grams per liter, or 300 grams per liter, this kinetics will alter depending on your initial substrate concentration. Yeah, and if you also start say where at time t equal to 0 you added 1 gram of enzyme, or if you start another experiment by adding 2 grams of enzyme, so your enzyme concentration will also alter the kinetics which you are observing here. Yeah? So like every chemical reaction, enzymatic reaction and their kinetics are in are how do you call, like influenced by different operating conditions. I'm going to list them, which is concentration of your substrate, and sometimes concentration of your product. Sometimes once you start to produce new products, it itself can affect your microbial, sorry, your enzyme kinetics. So sometimes your products can alter the kinetics of your enzymatic reaction, and how much concentration of your enzyme you added, enzyme concentration, suspension density, this is very important. Yeah? Suspension density can alter, temperature can change the kinetics of the reaction, and pH, depending on pH, the how fast you produce your product, that can alter. Yeah? So say if you have time, if you have pH, and say this is pH equal to 7 and pH is equal to 8, so pH is equal to 1, something like that. Yeah, so pH can also affect how fast you are producing the product per unit time. And then one more thing, so what are enzymes? So enzymes are like large protein molecules, just repeating this I think so, but still just take this like a message. They are large protein molecules composed by amino acids called polypeptide chains. I covered this before in the lecture 2, and also just now I said like this here I mentioned about this part. I think I talked about the alphabets of biochemistry in lecture 2, so try to have a look. So amino acids, they combine to form peptides, and peptides, they combine to form proteins. So I think we fairly covered some basics of enzymes. And one more thing, okay, why I said this part, like they are large protein molecules, and they call compiled by amino acids or polypeptide chain, yeah, composed by. So if you look into the chemical structure, it's not easy to draw a chemical structure in a notebook. For CO2 molecules, you can write C double bond O, or for water we can say like that. For water, and but the enzymes are large molecules. Yeah, I said to you 10 to the power of 4, which means you have so many number of atoms you cannot represent here. But if we look the chemical structure at atomic scale, so it will be like a like like a composed like a helical coils. Yeah, so like a ribbon. So we don't have to go into the structural details, but you should remember proteins have some unique structure, and it have this particular structure, and it will retain this structure because it can retain this structure only at some particular temperature and at particular pH. So if you alter the temperature or pH, so they will unfold. So these are like a folded molecules, so they will unfold, and then you will get some structure like that. Yeah, so you can call them as, I don't know, I cannot see that in the screen, so you can call these as unfolded proteins. So these are unfolded proteins, and these are proteins. So if you, let's me draw here, say this is your beautiful protein structure. So when you alter the temperature or pH, so it will lose its structural property, and then it will get unfolded. They call it as denatured proteins. And this can happen, that's because proteins have this structure because atoms are connected to each other to give this particular structure, but these connections are is due to some very weak van der Waals forces. So even if you slightly increase the temperature, you can break those weak bonds, and then you can completely rupture the structure, so you can unfold them. So, and it will lose its it will lose its it will lose its structure, right? It will lose its protein structure. So this diagram is a very simple diagram, but I talked about this because this is really important. So if you decided to produce some product from a substrate, the first thing you should look at its temperature and pH, because your enzyme can do lot of magic, like they can convert your substrate into products in no time, because enzyme have a possess this catalytic property, and they also convert more number of substrate to products per unit time. Yeah, so we will come to that particular topic. We define them as turnover number. Yeah, so I will I will talk about this particular property of enzymes, catalytic property, and we measure the catalytic property of enzymes using some parameter called turnover number. So when I'm going to talk about that one, you will understand. So, but one thing you should remember is like the enzyme can do its job only if you maintain its structure. Yeah, so that's why you have to give the right level of temperature. If the enzyme lose this structure and it becomes unfolded, then it anymore won't have the, so it will not do the job, so it will lose its catalytic properties. It cannot convert your substrate into products. And why is that? And there are so many reasons. If you lose the structure, so there is no catalytic property, and we will come back to that. Anyway, we are going to see some theory, or maybe we can call them as like a proven scientific hypothesis that will explain why this is happening, why protein structure is so important in order to perform an enzymatic reaction. And few more things you should, okay, one more terminology related to enzyme kinetics. I don't have to write. Yeah? It's called cofactor. So what is a cofactor? Imagine you have an enzyme, something like that. Let's call this as enzyme. And it won't, and if you add to substrate, it will not convert your substrate to products. But imagine you have one more molecule. We call this as coenzyme. Maybe I can change some nice color. Say this is your coenzyme, and when they combine together, say like, so this one, the combination of these two, they can convert your substrate into products. So in this case, we call this part as coenzyme, and we call this part as apoenzyme. Some people, they call it as cofactors. Yeah. So enzymes, like this type of enzyme, they can bound to a cofactor, and only in their presence, like this compound, they can so this is only this compound can convert your substrate to products. So, and this part of your enzyme, which is catalytically inactive in absence of your coenzyme, that's called apoenzyme. Yeah. And this unit, this where this inactive enzyme, which is bounded to an coenzyme and which have its and get the catalytic property, we call this as a haloenzyme. So these are some basic terminologies you should remember, and then we are going to see some important properties or maybe terminologies. Yeah. Anyway, we will see come to this. Okay, let me start. Turnover number. I'm going to explain this quickly. What is a turnover number? This is such an important number you have to remember when we talk about enzyme kinetics. But anyway, we will come back to this when you are going to derive Michaelis-Menten kinetics. I will derive this one probably in this lecture or the next one. So that time you will know what is the turnover number of an enzyme. But for the moment, I'm just going to explain or define. Yeah? So this number refers to the maximum number of substrate. This is a very important property or very important parameter. In fact, this is a property of an enzyme. Yeah? So that can be converted into products per unit time per active site of the enzyme. Yeah? So this is the sorry, I will take this one. Yeah. So this is the definition. So maximum number of substrate molecules that can be converted into products per unit time per active site of the enzyme. So this is something related to how many number of molecules of your substrate. So that's a number of molecules are converted into products per unit time per active site of enzyme. So number of active sites. Yeah. So again, that's a number of active sites. So this has a unit, something like a number per unit time. If time is expressed in seconds, so some number per unit seconds. So this thing refers to number of substrate molecules converted into products per unit time. So that's how you measure the turnover number. So this number like a, how do you call, like a characterize the catalytic property of your enzyme. So turnover number you can call, or you can take this as a measure of your catalytic performance, measure of your catalytic activity or catalytic performance. So what does that mean? So if this number is high, which means your enzymes are converting more substrate into products. If a number is less, which means your enzymes are converting less number of substrate, less molecules of substrate into products. So that's the meaning of this turnover number. And this number is normally very high for catalysts. Typically this number range from 10 to the power of 5 to 10 to the power of 11. So per unit seconds. So anyway, we will come back to this one. I will try to explain this way while we are going to derive the mechanism maintained kinetic expression. And then we need to know something about enzyme, which is their amazing specific activity. So what we are covering so far is like just the basics of enzymes. Yeah. So we are covering the properties of enzyme, what's the job of an enzyme, and where you get the enzymes. Like you break the cell and take the enzyme out, and then how they convert your reactants into products, and it has a catalytic property, it has some structural property, it has a definite molecular weight. So all we are doing is we are covering just the basics of enzymes. Even if you don't know what's the name of your enzyme, if you know it will do the job of converting your substrate into product, whatever I talked so far, that theory will fit. Yeah, like so which your enzyme is going to have all the properties that we discussed so far. And one more thing you should remember is specific activity. So enzymes are like they're highly specific. Yeah. So say like if this is your enzyme, how to write? Yeah. See this is your enzyme. So they cannot go and react with any reactant. Yeah. So they can react only with a specific reactant. Say like you have a, maybe let's put like this. You have reactant A, reactant B, reactant C, reactant D. They can act only with reactant A, not with B or C or D, because they are so specific. So enzymes are so specific to only a certain type of molecules, and so they can consume only molecule A and then convert them, they will act on molecule A and then they convert molecule A into products. So it won't even act on any of other molecules. Yeah? So they are so specific, and sometimes we call enzyme as like how do you call, absolutely specific. And sometimes we classify enzyme under group specific. So what does this absolutely specific enzyme mean? I will try to draw this. Yeah, this is going to be very tough. I'll try to draw this a bit neat. COO minus say this is your your substrate. Say this green part is your enzyme. So whatever the green color line you see, they are your enzyme, and the other one, the red color one, this one is your substrate. And you take the other one, I'll try to draw it a bit. This is very tough for me to draw. Say if you have like L and D form, we call them as chiral molecules or enantiomers. I hope you all know these things, because you are already in the third year. If you have these two type of molecules like L form and D form, your enzyme can act only on one form of your molecule, say L form, and that's because this enzyme have this specific shape of packets, or some active sites, so that can take only this form of your molecule. So only this molecule can fit into that space, and then this will be converted into products. And this one cannot enter that space, yeah, so no way. So the other form. So we call this type of enzyme that act only, how do you call, like that can recognize only one forms of chiral molecules. We call them as absolutely specific enzymes. Yeah. So, and then what is group specific? So we all know. So I'll try to write here. And another one is so this is methanol. So this one is ethanol. Imagine if you have an enzyme of this shape. So this blue boundary is your enzyme. And just assume that enzyme have some packet that can take only this particular group. This part of the group, yeah. So this one. And again here. So some enzymes, they can, how do you call, like they're specific to, like these are closely related if you take only this part of the molecule, yeah. So enzymes that can recognize like closely related molecules or substrate, or that can recognize molecules that contain some specific type of functional groups, we call them as group specific. So in this case, enzymes can take both this type of compounds and then they convert them into products. So we call that type of enzymes as group specific enzymes. I hope you all are following this one. I'm just checking my notes where cover turnover numbers, specific activity. I think covered almost everything so far, I think. Probably no, one more thing, which is a specific activity. specific activity of enzymes. So how to measure the activity, specific activity of enzyme, yeah? So there is no units to measure the specific activity of an enzyme. So what does this mean? Like I already said, what is this catalytic, this turnover number and something, yeah? Like so how do you call, like so how to say it simple, like you take your substrate and then you add your enzyme and then you convert your enzyme into products, yep, and then try to plot that one. You have your product concentration on y-axis and time on your x-axis, so you just plot it here. So what it says is like you're producing some products, yeah? So this is something like a change in product concentration with respect to time. So there is some, if you calculate the kinetic constant, you will get mass of product produced per minute. So just imagine you start an experiment, time versus your product. So you convert your product concentration is going to increase, yeah, with respect to time, and you do another experiment and your product concentration is going to increase, and just imagine you do another experiment and your product concentration going this pattern. You take the slope of here, the initial region where you see some linear line, so that will give some number that will calculate, like if you take a slope that will give mass of products produced per unit time, yeah? So we call them as activity of enzyme. So coming back to this specific activity of enzyme, so the unit for specific activity is unit, believe it or not, that's how they call it's funny to me, I don't know why. It's the unit for specific activity of an enzyme is just called units. That's how the biology people, they, I don't know, they fix the unit for this one, and there is a reason for that, you know. Probably they have some practical difficulties to define a definite unit. Like for concentration, you can tell the unit is grams per liter, moles per liter, or like, so or if you take a like weight, you can tell in grams, milligrams, or kilograms. But how much products are converted per unit time by your catalysts, that's very difficult, yeah. So to fix a unit for that one. And why is that? And that's because I said to you before, you have a live cell and then you have something inside, we call them something inside, we call them as enzymes. So when they manufacture enzymes at industrial scale, they produce a lot of these cells, and then they break the cell, and then they take the enzyme, yeah. So and then they try to purify them. So when you break the cell, you will also have some other compounds, maybe let's put new color, some other compounds inside the cell that may not have this catalytic property. And once you break the cell and you take out, you will get the cells that contains the catalytic, sorry, the molecules that have the catalytic property, and also the molecules that does not have the catalytic property. So what they people do is they purify them until they get very less number of this the molecules that has no catalytic property and more number of enzymes, say this one, and you further purify this, and then you will get say near to 100% pure enzymes, yeah, pure enzymes, and almost a negligible amount of molecules that has no catalytic property. So this is like less pure. Say we can call purity of 80%. Say this one purity of 90%. Say this one purity of 95 percentage. So this one contain 95 percentage of just enzyme that has the catalytic property, and 5 percentage of the molecules that you get from the cells that may not possess any catalytic properties, yeah. So this is the this is how you manufacture enzyme, yeah. So normally this will be dissolved in some suitable solvent. So normally if you want to call the concentration, like milligrams of all the molecules inside, sorry, milligrams of your all the molecules per unit liter of your solution, yeah. So you cannot just cut with the normal scissor, yeah. So they all are dissolved in solution, all this. So that's why, so this is something like milligram of your catalyst per liter, milligram of catalyst, milligram of catalyst per liter of solvent, if you want to call. And just imagine, let's change the color back. You do one experiment. Say you take your time, your product, you take your substrate, and then this time you add 80% pure enzyme, say some X grams of your enzyme, and then you do say 1 gram of enzyme, yeah, and 1 gram per liter of enzyme, or 1 gram of enzyme, let's put like, and then you monitor how much your product is changing with respect to time. Say this is with 80% purity, and then you do one more experiment with 90% purity, 90% purity enzyme. So the next experiment you add 1 gram of enzyme, and third experiment, sorry, 1 gram of enzyme of 90% purity. You do third experiment 1 gram of your enzyme with 95% purity. So you will get results something like that. So now you take the slope. If you express this in terms of milligrams per liter, so now we can define the activity like milligrams per liter per unit time. So this is your activity for this one. This one will be less than milligram per. So this will goes to 90%, and with 95 percentage your activity will be much higher, yeah. So that's the meaning of that. And imagine here I use the unit like 1 gram of enzyme. Normally enzyme will be dispersed in some solution, so you can say like 1 gram per liter of that solution, yeah. So and you divide this value by, or if you are going to express the unit of enzyme just in grams, you can unit that's you can express, why not, you just divide everything by gram of your enzyme, gram of your enzyme, gram of your enzyme. So this will give your specific activity. And they call this as unit, just unit. So unit of specific activity is just unit, and that unit will be that unit will be expressed in terms of like milligrams per liter per time per gram of your gram of your enzyme. Milligram of products formed per unit time per gram of enzyme. and this value is going to change depending on the purity, of your enzyme. And, and since you will never know how many grams of pure enzyme and how many grams of other molecules that you get while you break your cells and extract your enzyme, so this, nobody will know that, like whether this is 100% pure or not. So that's why they try to call us units. And in fact, the only way to check your purity of your enzyme is to perform a series of experiments with the same substrate concentration. You purify one time your enzyme and then calculate P versus time, and then you do one more time, another time, and another time, and another time. You repeat that experiment until you see there is no change in product concentration with respect to time. Like, so this one refers to pure enzyme. This is a bit very tricky. This is such a simple process or a simple definition, but this looks tricky because this is connected with enzyme purity, and enzyme purity is a very tricky parameter to obtain from experiments. So normally, if you really want to compare the purity of enzyme, the best thing is to go to lab, take your substrate, convert it into product using same mass of your catalyst, and then see which catalyst convert more products per unit time. So that's the best way. So that's how you check, that's how you test your activity. And once you divide that activity by gram of your mass, then it will give you something called specific activity. So we will read more about activity, specific activity, and turnover number. So we will cover this one in the next lecture in detail. So while we are going to maybe we can try to solve a few problems that will help you to understand what's the how do you get activity if you have an experimental data, how do you get specific activity, that because you need to know specific activity. That's one way to characterize your enzyme. So I will try to solve few problems, so that will be the easiest way to learn this concept. Okay, I can do one problem now because I have this already in the notes. So one more thing you have to remember, this one, time, your product concentration expressed in, say, millimoles per liter. Imagine if you take an enzymatic reaction, you add substrate, add enzyme, and then you monitor your product concentration using some probe, some spectroscopic technique. If you get something like that, the initial region where you have something linear, we call them as initial velocity. So this change in this slope value, or you can call this as just activity. So just I'm going to call this as initial velocity in chemical engineering terms. So if you take a slope, that will be equal to millimoles per liter by time. If time is expressed in seconds, it will be equal to millimoles per liter per second. I hope you all know how to take a slope. Millimoles per liter per second. So this is your initial velocity. Now let's try to solve the problem. So we performed some biological enzymatic reaction, and we we got some result like this, product versus time, and we take a slope of this initial linear region, and then we found or we calculated that initial velocity. So we can call, so let the initial velocity, or you can call this even as a reaction rate. Initial velocity is equal to 3.08 millimoles of products per liter per second. So calculate, we added some mass of enzyme while we perform this experiment. So we added, say, like you started this experiment with 0.0001 millimole of enzyme. So calculate the specific activity. Calculate the specific activity of enzyme. Yeah? So specific activity is just your initial velocity so your specific activity, initial velocity divided by mass of catalyst. So that will be equal to 3.08 millimole per liter per millimole of enzyme. This one is millimole of product. So this is it by 0.0001. So whatever the answer you get, this will be equal to 3800 millimoles of products per liter millimole of enzyme. So this is your specific I hope. Yeah, so this is your specific activity. Let me write it here. So in the end, in the exam, if I give you some number and ask you to calculate the specific activity of your enzyme, or if I give the specific activity of enzyme and if I give the mass of catalyst, you should be able to calculate the initial velocity. Yeah? So try to have some understanding on this simple equation. This is really important. Yeah. So I think I'm going to stop now in the, or maybe I can try to cover a few more topics in this lecture. So the next lecture I can just focus on the mathematical derivation part. Or maybe I will stop now. Yeah, it's better. I will in the next lecture. I'm not going to use YouTube. In the next lecture, I'm going to record it directly on the PowerPoint because I need to show you some GIF images. I cannot show in this paper tube. It's not allowing me to upload a GIF image or even a normal image from internet. So next lecture will be recorded directly on the PowerPoint. I'm going to stop the recording now. So next lecture I'm going to cover, like in this lecture I said the enzymes are so specific and it can act only on like one type of your enantiomer, or it can act with, or it can recognize molecules with certain specific functional group, yeah, certain functional group. So I will tell the reason why is that, and there are few theoretical hypotheses for to explain why enzyme recognize molecules in such a pattern, yes, in such a way, sorry. And then I will also talk about a bit about the stability of the enzyme, and I will also, and then I will try to derive Michaelis-Menten expression. This is that's a very important expression in bioprocess engineering. Yeah? So I know many of the products that are produced using enzymes. If you want to study the data, or if you want to gain some theoretical understanding, or if you want to gain some engineering level understanding, then you should know the meaning of mechanism and kinetics. And the enzymes also have some property, like if you want, you can stop the activity of enzyme using some inhibitors, and you need to know like what type of inhibitors you are dealing with. So for those things, like you need some mathematical expression. So those are the things that we are going to cover in the next lecture. So this enzyme kinetics is maybe tough for some of you, especially who do not have engineering background, because other than these definitions which I discussed in this particular lecture, we are going to have a lot of a few more derivations and some mathematical expressions which you need to use to solve some problems. So next lecture I will try to complete everything in enzyme kinetics, and after that I will try to solve a few problems. I heard Jack already started to give you some tutorial using from the science learning center. So I will talk with him this week, and I will ask him to solve a few problems related to enzyme kinetics in the next week. So but I hope you all are reading whatever I'm posting in the YouTube, or also in the PowerPoint, because if don't leave it until the last minute, because there are so many things you have to remember, and in your final exam you might lose the track, because I'm going to ask some problems, and you should know which equations to use in order to solve those problems. So try to read immediately, or if you have any doubts, you can ask me. Thank you. I'm going to stop the recording now. I will upload it in the Solis immediately. So just remember, the next lecture will be directly recorded on the PowerPoint. Thank you.